Exploring Life, Chapter 6. Origin of Life. Part 2. Life appeared after the planet had formed, but before the rise of massive stromatolite mats. So the big question is, how did it begin? There are two main theories that attempt to explain the origin of life. The first is the panspermia theory, which suggests that life, or at least its building blocks, arrive from space. This could have happened via meteorites or comets carrying microbial life or organic compounds to early Earth. The second is the abiogenesis theory, the idea that life originated on Earth through natural chemical reactions. In this view, non-living molecules gradually assembled into self-replicating systems, eventually giving rise to the first cells. As of now, both theories remain scientifically plausible, and researchers continue to investigate evidence supporting each. Let's take a closer look at the evidence for panspermia. One line of evidence supporting the panspermia theory comes from the discovery of organic compounds in meteorites. Scientists have found amino acids, hydrocarbons, and even nucleic acid components in space rocks, compounds that closely resemble those found on Earth. This suggests that some of the building blocks of life may have come from extraterrestrial sources, lending support to the idea that life, or its ingredients, may have arrived from space. But it raises an interesting question. What if it wasn't just chemicals? Could living organisms actually survive the journey through space? Surprisingly, the answer might be yes. Some extremophiles, like tardigrades and certain bacteria, have survived conditions that mimic the harsh environment of space, including radiation, vacuum, and extreme temperatures. These findings suggest that it's at least biologically possible for life to endure space travel, adding another layer of support to the idea that life on Earth could have had an extraterrestrial origin. However, it's important to remember that panspermia doesn't explain the ultimate origin of life, only how it might have arrived on Earth. Wherever it happened, here or elsewhere, abiogenesis, the emergence of life from non-living matter, must have occurred at least once. And understanding that process is key to understanding life itself. The real question isn't just where life began, but how it emerged from non-living matter. We don't assume life is eternal, because we believe the universe itself had a beginning. So at some point, life had to arise from non-life. Whether you're inclined to believe in panspermia, that life or its ingredients came from space, or in an Earth-based origin, the core challenge remains the same. How did non-living chemistry become biology? Whether it happened on Earth or elsewhere, the process of abiogenesis, the emergence of life from non-living matter, had to happen somewhere. And understanding how that happened is key to understanding life itself. Before diving into the possibilities outlined by science, let's take a moment to reflect on a question that touches biology, philosophy, and belief alike. Where do you think life on Earth originally came from? Was it formed here on Earth, through natural chemical processes? Did it arrive from space, carried by meteorites or comets? Or could it be something entirely different? Some speculate about alternative origins, such as life being seeded by an advanced civilization, or even the idea that we live in a simulation. And for many, the question of life's origin is deeply tied to religious or spiritual beliefs, seeing life as part of a greater design or purpose. So, what do you think? In this course, we'll focus on examining one possible path for how life might have arisen from non-life, right here on early Earth. This slide outlines 4 major stages in the transition from chemistry to biology. Let's take a look at them in a more visual form. This schematic illustrates how each stage may have unfolded, step by step, from simple molecules to the first self-replicating systems. First, the formation of organic monomers. Simple molecules combine to form the basic building blocks of life, like amino acids and nucleotides. Second, the formation of organic polymers. These monomers linked together to create more complex molecules, such as proteins and nucleic acids. Third, the formation of membrane enclosures. Membrane-like structures emerged, allowing early life forms to separate their internal environment from the outside world, a key step towards cellular life. And finally, the emergence of self-replication. Certain molecules developed the ability to copy themselves, making heredity, variation, and eventually, evolution possible. Now we'll examine the evidence supporting each of these steps, and let you decide for yourself whether abiogenesis on Earth seems like a real scientific possibility. Let's begin with the first stage in the appearance of life, the formation of organic monomers. This stage is often explained by the primordial soup hypothesis. According to this idea, organic monomers, the basic building blocks of life, like amino acids and nucleotides, formed from inorganic compounds present on early Earth. These monomers likely accumulated in the ancient oceans, creating what scientists call a primordial soup, a rich, reactive mixture that set the stage for the next steps in the origin of life. In 1953, American graduate student Stanley Miller conducted a groundbreaking experiment that brought the primordial soup hypothesis to life, literally. He attempted to recreate the conditions of early Earth's atmosphere in a laboratory setting. The experimental setup included a mixture of gases believed to be present at the time: methane, ammonia, hydrogen, and water vapor. By simulating lightning with electrical sparks, Miller tested whether simple chemical reactions could produce the building blocks of life. To simulate early Earth conditions, Miller applied electrical sparks to the gas mixture, mimicking the effect of lightning in the primitive atmosphere. The electrical energy helped trigger chemical reactions between the gases. After several days, organic molecules, including amino acids, began to appear in the liquid solution. This result provided strong support for the idea that life's building blocks could form naturally under the conditions thought to exist on early Earth. This schematic shows the setup of the Miller experiment, designed to simulate conditions on early Earth. On the right, a boiling flask contains water, representing Earth's ancient oceans. Water vapor rises into a reaction chamber on the left, where it mixes with gases like methane, ammonia, and hydrogen, thought to mimic Earth's early atmosphere. Electrodes deliver electric sparks, simulating lightning storms from that time. These sparks provide the energy needed to drive chemical reactions between the gases. The resulting mixture circulates through a condenser at the bottom, which cools the vapor, causing it to condense and collect in a trap. After several days, the liquid in the trap contained organic molecules, including amino acids, the basic building blocks of life. The Miller experiment was a breakthrough in our understanding of life's origin. It showed that organic monomers, like amino acids, could form from inorganic substances under conditions similar to early Earth. This was the first experimental evidence that life's building blocks could arise naturally, without any biological system. Our understanding of early Earth's atmosphere has evolved, mainly in the proportions of gases. So the experiment has been repeated many times. And the result is always the same. Organic molecules still form, including amino acids, nucleotides, and simple fatty acids or phospholipid-like compounds. Life's ingredients can emerge from non-living matter. The second stage in the appearance of life is the formation of organic polymers. In modern organisms, polymers like proteins and nucleic acids are synthesized inside living cells, with the help of enzymes and complex machinery. But in the early Earth, cells didn't exist yet. So how could polymers have formed? Some hypotheses suggest that under the right conditions, organic polymers could form outside of cells, without the need for biological systems. This step would have been essential for building the molecular complexity needed for life to emerge. One hypothesis for how organic polymers may have formed without cells is the clay model. In this scenario, organic molecules could have assembled on non-organic clay surfaces present on early Earth. Dehydration and rehydration cycles, driven by tidal movements, generated by the Moon, may have helped link monomers into longer, more complex chains. Additionally, the charged surfaces of clay particles may have acted as natural catalysts, helping to organize and accelerate the chemical reactions needed to form the first polymers. And this isn't just theory, lab experiments have replicated key aspects of this process. Experimental evidence supports the idea that clay minerals could have played a role in the origin of life. Laboratory studies show that certain clay surfaces can catalyze chemical reactions, helping simple molecules link together into more complex ones. Wet-dry cycles, similar to those caused by tides or evaporation, further promote the assembly of polymers, especially when combined with chemical exposure. Together, these findings suggest a plausible scenario for how organic polymers might have formed without any living systems, offering a key step in the path from chemistry to biology. The third stage in the appearance of life is the formation of membrane enclosures, structures known as protobionts. This was a key step toward the development of the first true cells. In water, certain chemicals, such as phospholipids, can naturally form small, enclosed bubbles similar to soap bubbles. These enclosures could trap whatever organic material happened to be nearby. Some of these tiny bubbles showed basic stability, and as they drifted away from their origin, they maintained a clear boundary between their internal contents and the surrounding environment. This simple encapsulation was important, not because the contents were organized, but because they were protected and concentrated. It was a critical step toward building the complexity needed for life to eventually emerge. Recall from the previous chapter, in water, modern cell membranes are built from phospholipids. In the bilayer that surrounds each cell, the polar heads, which are attracted to water, face outward toward the watery environment, while the hydrophobic tails, which avoid water, are tucked inside. It might look like a complex structure, but here's what's astonishing. Phospholipids naturally form this bilayer. They self-organize because of their dual nature. When shaken or agitated in solution, phospholipids can even spontaneously form vesicles, small, enclosed spheres surrounded by a bilayer membrane. So, early protobionts may have had the same kind of membrane wrapping as modern cells, providing a stable environment for trapping and concentrating organic molecules. This was a critical step toward the formation of the first cell-like systems. As protobionts formed, they created enclosed spaces where key molecules, like nucleotides, could become concentrated. In modern cells, RNA carries genetic information, but it can also act as a catalyst. These catalytic RNAs, or ribozymes, can speed up chemical reactions. Even modern ribosomes rely on RNA for catalysis, showing RNA's lasting importance. Inside protobionts, nucleotides may have polymerized into RNA strands, some of which began to fold and function as catalysts. This suggests RNA played a central role in early metabolism, laying the foundation for the transition from chemistry to life. The timeline of ATP's emergence, the energy currency of modern cells, remains unclear. ATP closely resembles an RNA nucleotide, specifically adenine, which supports the idea that it may have evolved from earlier RNA-like molecules. Though a variety of more primitive energy carriers may have come before it, ATP eventually emerged as the universal energy carrier, likely due to its efficiency and versatility in powering biochemical reactions. The fourth stage in the appearance of life is the development of replication, the ability to copy information and pass it on. As protobionts continued to evolve, short RNA molecules may have formed through polymerization, creating primitive RNA genomes. Initially, environmental factors such as temperature shifts, chemical gradients, and pH changes may have caused protobionts to split into smaller vesicles, dividing their RNA content by chance. This unorchestrated process, driven purely by environmental forces, foreshadowed cellular reproduction. While it lacked precision or inheritance, it marked an important step toward the growth and division behaviors we associate with living cells today. After protobionts incorporated RNA, they began to replicate, mutate, and evolve more reliably and predictably, gradually acquiring more and more properties of life. Over time, these systems came closer to meeting the scientific definition of living organisms. The RNA world, a period when RNA molecules played a central role in the earliest lifelike systems, is considered the transitional stage between non-life and life, taking place inside protobionts. In early evolution, RNA served dual roles. It could store genetic information and also act as a catalyst for chemical reactions, something no other single molecule could do as effectively on its own. And eventually started to play the role as energy currency. This set the stage for natural selection to take hold, favoring molecules and systems that were more stable, more efficient, and more capable of passing on their structure. Through this process of molecular refinement, the first rudimentary genetic and metabolic systems began to stabilize. The emergence of protobionts and the rise of the RNA world marked a critical transition from non-living chemical systems to the first forms of cellular life on Earth. And here's something remarkable. Since all known cells share fundamental features, like genetic code, membranes, and core metabolic pathways, it's likely that only one successful combination of molecules gave rise to all modern life. Once that system emerged, it outcompeted all alternatives and became the foundation of the tree of life, a single origin from which the vast diversity of life on Earth evolved. Now it's time for review. Feel free to take notes and move back and forth through the slides as you answer the questions. Don't rush, take your time, and really reflect on everything you've learned.